So thank you very much, Kerry and Tony and Benoit, for this nice invitation, and to SAMS and QUT. So this is a joint work with Pavlo and Myriam, who work in France at Telecom Paris and Crest and Sae, where I was colleagues. I was a colleague of these two persons 2 years ago, and in fact. This is a work that started when I was in Sydney. So at the time I was working at the University of Montreal, and I had a sabbatical, and I spent 1 year working at the hospital in the School of Psychiatry at UNSW Sydney, and I was working on these interesting data sets. And this was where everything started, basically. So what was it about? It was about trying to explain something about the white matter. So white matter is something in the brain where we look at the axonal fibers, which are fibers that relate groups of neurons to transmit information between these groups of neurons. And the goal of the project was to try to see if there was some kind of genetics involved in the white matter integrity. All right, and this is interesting from a medical point of view because if genes are involved or genetics is involved, then we have to treat using gene therapy. And if genes are not involved, then we need to look at what are the environmental factors. So it was the OZ study, so a big study on old Australian twins. And these are the kind of information that we have. And here I will only focus on the zygosity, which is are the twins, identical twins or non-identical twins, and the MRI measures, so the information of images in the brain. So how do these things are measured? Basically, so these fibers, the water molecules that are inside the brain, they move easily along the fibers than in an orthogonal direction for these fibers. So we can track the displacement of water molecules using what is called diffusion tensor. And we obtain these DTI images, diffusion tensor images, and we can track the fibers using that kind of information. All right, so what I studied was the main fiber tract in the brain, which is called the cerebrospinal tract, which goes from the brainstem to the motor cortex that deals with movement of legs and arms and stuff like that. All right, so these are the data that we have, basically, and yesterday you were able to kind of plot these data. And we need some sort of modeling of these data. So these data are fibers, curves, and what can we do to try to model these things? So there is so when I was in Brittany, a few years after that project started, Pavlo came and he was working on this notion of half-space depth, right? Which is something that describes the centrality of a point with respect to a multivariate cloud of points. All right? And this can be extended to the depth of a point with respect to a probability distribution also. So if we have a distribution Q on the Euclidean dimensional space, and we have a fixed given point X, we want to be able to compute the centrality of this point X with respect to this measure Q. And there is the equivalence of trying to find the depth of this point X with respect to a cloud of points, right? And this is defined as the infimum of the probability of this half space here for all these half space that goes through X, basically. So I did this figure so that you can understand a little bit better what I'm talking about. So this is when G equals to 3 in 3 dimension, basically. So we have this point X here. We have this vector U here, which lies on the unit sphere. and it's difficult to see the sphere here a little bit, but anyway. And then we have this half space HU of X, which is the space which is on the right of this red half plane, right? So in 2 dimensions, if we want to compute the centrality of a point with respect to a cloud of points, this is how we proceed. So we have this point X here in red, which is given, and we want kind of compute, is this point in the middle of the other points, or is it on the outside of the other points? So as I just explained, I'm going to look at all the half spaces that go through that point, and then I'm going to compute the probability of the points that are within that half space. All right? So here we have, in total, we have 161 points. So the probability, kind of probability of this half space is 120 divided by 161, right? And then we move all these half spaces, and we consider all these half spaces, and we try to find the infimum as given in that formula above here over all those planes, right? So this is the infimum, and this tells you if the red point is within the depth of this point, basically, right? We can consider another point here. So you can see that this point is more on the outside, and I can do the same job, basically, moving around, right? And then I'm trying to look at the infimum, which is here, right? So this number is small, so that tells me that the red point is well on the outside of the cloud of points. All right. So now, can we use that kind of notion for curves? Well, the points we have, when we observe these curves in the brain, of course, every observation, physical observation, is made of points, right? So the brain is divided virtually in small volume elements that are called voxels, and we have the location of the curve along these voxels. And so we could think about using this data depth notion on the points that are on the curve, but this is not a good idea because we would lose the the link between points on the same curve. All right, so can we adapt this original concept to something more advanced? So there is a version of depth for functional data, right? So the idea is just we consider this stochastic process, some sort of realization, one realization of it, and then we compute the original data depth for each point on this curve here with respect to the other one, and then we integrate all that things, right? So can we use that notion for our curves? Well, our brain fibers live in a 3D space, not in a 2-dimensional space, right? So we cannot use directly that notion. Well, some people worked in 2014 to a multivariate extension of this, right? So it's kind of a direct extension. And can we use now this multivariate functional data depth on our curves? Well, I'm going to try to explain why using functional depth is not a good idea in our context. So say we have these curves, these shapes here, and we are interested in computing the centrality of the red curve with respect to the black curves, right? So we can consider 2 different parameterizations of the above curve, right? So parameterization A and B. And in fact, these 2 parameterizations lead exactly to the same black curve, right? So this is kind of explained here. And if we compute, so I can maybe show this here. So this is the first parameterization. So you can see that the blue point goes slowly first and then goes more rapidly, right? And if I look at the second parameterization, then it goes quickly first and then slowly afterwards. But these 2 parameterizations draw exactly the same curve, basically, all right? And if we compute the functional depth on these curves using one of the 2 parameterizations, then the depths are different, all right? We did that, for example, on this classical FDA data set. And we can see that the rankings here for two different parameterizations, so parameterization by time or by arc length, are completely different. So for example, this one is ranked number 1 for this parameterization, and this same curve is ranked number 19 for the other parameterization. All right? So using this notion of functional data depth for curves is highly dependent on the parameterization that is chosen for the curve. All right? And for the data that I'm dealing with in the brain, we don't have any natural parameterization. So we need to create something different. All right. So I need to introduce a few concepts about parameterized curve and unparameterized curve first. So what is a path? A path is just a continuous map from the 0-1 interval to some Euclidean space, right? And we are going to note S gamma the locus of this continuous map, right? So we start from some point 0, we are at A, gamma of 0, and then we go along that curve until we go to the point B, right? And then because we want to get rid of these parameterization that lead exactly to the same curve, what we are going to do is to use, to define an equivalence relationship between two curves, and we will say that these two curves, gamma 1 and gamma 2, or two curves, gamma 1 and gamma 2, are equivalent if they share exactly the same locus. So when they define the same curve in space, and the points on the curve are visited exactly in the same order. All right? And also, the particle that moves along a curve never stops. All right? So when we do that, so I already introduced that. So when we do that, we can define what we call an unparameterized curve, which is just an equivalence class on the set of all curves. Right? So we consider that two curves are equivalent, are in the same equivalence class, if they share the same locus and visit this locus in the same order. Right? And then we can define from there, we can define the space of unparameterized curves as being the set of all these equivalence classes. All right? So we can define, so this is gamma, our space of unparameterized curves. All right? So we can define a distance on this space, and then thanks to the distance, then this space becomes a metric space, and we have some nice theoretical properties that were used in the various proofs that we have in the paper. All right? We have to define also the length of a curve. So to define the length of a curve, basically we just take a partition of the 0-1 interval, all right? And then we compute the length of each red segment within each small bit of the partition, and then we add the length of all these segments. So we sum all the length of these segments, basically, and we consider the supremum over all possible partitions. And when we do that, this defines the length of this curve, okay? Because if we define a partition which is finer and finer, then this will approximate better the length of this black curve. And we will consider rectifiable curves. That means curves that have a finite length. We have something also called the normal parameterization, which is a way to parameterize the curve using the so-called arc length parameterization, where the particle that would define a curve would move at a constant speed. All right. Now, we have to define what we call what is called the line integral of a function over a curve, and this can be useful for various reasons. And basically, then we introduce a probability measure associated to a given curve C. Remember, the C is a class equivalence so parameterization are lost in this object here. And so basically, it's just the line integral of this function here over some set A in the Borelians, and divided by the length of the curve, right? So if I have, for example, this set A, then this part here is just the length of the light blue portion of curve here, divided by the total length of the curve. So this is how we define this probability measure on the whole Euclidean space. All right. So it's some sort of uniform measure, but which is extending from the curve to the Euclidean space. All right. Then this is our statical model. All right. We consider all the probability measures on the space gamma, which is a space of unparameterized curves, which have a finite length. And we consider that the curves that we observe are IID, identically independent, identically distributed and independent, from some probability measure that is taken on this space, right? So this kind of generate curves according to this probability measure. And then we have to define a probability measure QP on the Borelian of the Euclidean space by kind of computing the expectation of some random element on this space of curve. All right. And of course, in practice, we will have to compute this data depth. I will introduce the definition on the next slide. So we will need some kind of Monte Carlo estimation sampling scheme. So basically, we will generate observations. So the curves lie in a three-dimensional space. We will generate points in the three-dimensional space that will be on these curves, and such that the distribution of these points given some curve is the distribution mu that I just introduced on the previous slide. So they are uniformly generated along the curve, basically. So we can illustrate this like that. So we have these blue curves that we observe, and say we want to compute the centrality of the red curve with respect to the blue curves. All right. So we are going to generate observations on all these curves, and then we are going to use these observations to try to compute the centrality. Right. So this is our definition of the curve depth of one given curve C, which is the red curve here, with respect to a probability measure P defined on the space of unparameterized curves. And this is some sort of equivalent of the definition that I introduced on the first slide when we had these moving half spaces in two-dimensional space, right? Remember, I was moving around all these things, right? So I can just quickly go back to this. So this is what I did, right? So I want to generalize this definition here, the infimum on the probabilities on half spaces, or this one for an empirical version of that, to the space of unparameterized curves. So this is what we did. We defined this Turkey curve depth with respect to a set of curves as the infimum of the probability distribution of a half space divided by this thing, right? So intuitively, this is how it works. We have these blue curves. We want to compute the centrality of the red curve with respect to the blue curves. We consider this half space here, and then we will consider all the half spaces that go through that point, all right? And we will divide, we will compute this ratio here, and this ratio is what? So QP of HUX is measuring, in fact, the expected fraction of the length of this red curve. Of the length of a curve that would be generated by P that would delve into this half space, right? And then we will divide by the fraction of the length of the curve C, which is the red curve that delves into this same half space. Right. So this works like that on a very simple example. So we have these curves. One computes the centrality of the red curve with respect to the blue curves. We generate points on these curves, and then we consider a half space. So this is a half space above this line here. Of course, in our context, this will not be in two-dimensional space, but in a three-dimensional space. And then we compute this quantity, which is so at the bottom here, we have the fraction of length of the red curve that goes into the half space. So we have 1, 2 points over 1, 2, 3, 4, 5, 6, 8 points. So 2 over 8. So this is a fraction of the total length that goes into that half space. And we do the expected fraction for all the other curves, right? So we have 5 other curves. So we compute this fraction of each curve that goes into that half space, and then we take the average of these fractions, all right? And we do that for different half spaces, all right? So same thing for this half space, same thing for this half space, and we compute the infimum over all half spaces, all right? And of course, we have a computing code that does that for our curves, and it does that, you can imagine, in a three-dimensional space where we have to move around all these half spaces in all the directions, all right? And each time compute this quantity and try to find the infimum, right? And first we have to generate a certain number of points on each curve. So it's quite time-consuming, in fact. All right, so I will not go into all the details of the proofs for that. So we have this paper on the archive if you are interested. And basically, we were able to prove a few results, like this new notion of data depth for curves is always between 0 and 1. We have a similarity invariance. So if we rotate the whole bundle of fibers, for example, or scale the whole bundle of fibers, then the measure of centrality of a given fiber does not change, and vanishing at infinity. So if we take a curve and we send that curve far away from the other curves, then of course the measure goes to 0, all right? And we have an empirical version of that we use, and we prove the consistency of this empirical version. So we applied these things to our brain fibers, all right? So remember what we have. We have these Australian twin studies, study where we have all these diffusion tensor images for 34 twin pairs. All right, so we have 11 twins, 11 pairs of twins that are non-identical, so these DZ guys, and 23 pairs of twins that are identical twins. All right. And from their brain images, we extract using this fiber tracking thing, we extract all 1,000 fibers for each person on the left hemisphere and on the right hemisphere that goes from the bottom of the brainstem to the motor cortex. And we have a few questions that we would like to address using our new technology. The first one is information compression. So the idea is to try to replace this bundle of 1,000 fibers by just one best representative of this bundle of fibers. So just replace 1,000 fibers by just one fiber, which is the best representative, the median curve, if you want. We would like to be able to detect outliers, because sometimes when we do the tracking, there are some problems with this algorithm, and we would like to track to detect these fibers that was not correctly tracked. We want to do what is called in neuroimaging curve registration. So curve registration is the fact that you have several different people, and you want to align all the bundles of all these people in the same space. All right? So you want to rotate basically all the bundles of fibers of everybody so that they are all aligned in kind of the same direction. So this is called curve registration. And the last one is to study the genetic dependency. So here it's more something called the heritability. So we do not use the genes or the SNPs in that context, but just the fact that we know that identical twins share 100% of the genes, whereas non-identical twins share only 50% of the genes. So these are the data that we have for just one subject, and we applied the functions that we have developed to these fibers, right? And so this is what we got. So these are the one-size fibers. I think there are only 100 represented on the left and on the right, so that's I can move this thing around smoothly. And so this is a way to visualize. So this is interesting also for the neuroscientific people to be able to visualize a bundle of fibers by removing the external fibers, right? So doing that, you can see the blue curve is the curve that is right in the middle, right? The more central curve. And this we have the same thing for the other bundle, all right? Okay, so we computed the depth of all the curves, right? So for each curve, we have a number between 0 and 1 that tells us if the number is 1, then the curve is right in the middle. If it's close to 0, it's on the outside of the bundle. So we have this number between 0 and 1 for each fiber. So doing that, we can choose the fiber which has the highest number, and this is our median curve and the most representative element from the bundle, right? We can do outlier detection, all right? So this for one subject, this was the bundle of fibers, all right? And then if you draw in a different color the fibers that have a depth which is very small, so this is what we did. So the red curves are the curves that have the depth which is very small, very close to 0. And then we can see right away that these fibers are outliers, all right? So of course, when you have just a few people to deal with, you can display this bundle of fibers and look at them and identify manually the fibers that are problematic. But when you have a lot of people involved in the study, then you cannot do that for every people because it becomes very time consuming, right? So you have to have some kind of algorithm that is able to detect the outliers automatically and remove them to go further in your analysis. So we can put some kind of threshold, for example, and get rid of these outliers curves. So curve registration, so we want to register all these 68 brain bundles together. So for example, on the right hemisphere here. So what we did, we took all the 68 people, we took their right bundle of fibers, and for each people, we computed all the depth of these curves, and we extracted only the middle one, right? So for each person, we ended up with this thing, basically. Then we put together all the middle fibers of all the 68 people. So we came up with a new bundle of fibers of the deepest fibers of everybody. And then we computed the deepest of all, right? So the deepest of all was for this subject 235, right? And then we want to realign, spatially align all the bundles of fibers for every subject to this subject 235, right? So the blue-black curve here, this is the more central fiber for this subject. And then we consider all the other subjects one by one, and we consider their more central fiber. So for this subject, the red curve is the more central fiber of the bundle of this subject. And we try to align this red fiber to this fiber here. Right? And to do that, what we did, we computed the distance, the Fréchet distance I mentioned on the previous slide, and we try to minimize the distance between these two fibers. All right? And doing that, for example, we were able to reduce, so for this guy, for example, the original distance was around 10, it was reduced to 3, and this one it was 4 something, reduced to 3, and 3 something, reduced to 2, right? So this was a way to realign all the bundles of fibers for all the persons. And once we have that, we have all the bundles aligned spatially. We then computed what are called these DD plots. All right? So the DD plot is we take each fiber of a person, for example, this red point, red dot here, red circle, it corresponds to one fiber of this person 11029, and we compute the depth of that fiber with respect to all the fibers of the same subject and with respect to all the fibers of the other subject. The idea being that if the two subjects if the fibers generated for the two subjects are independent and identically distributed, removing one fiber and put it in the other set should not make a difference, right? And so we did that for every fiber, for every person, for the 68 persons, 34 pairs, and then we plotted this kind of plots. And you can see here, so DZ, remember, these are the non-identical twins, and MZ, these are the identical twins, right? And then we can see right away that we can see by the eye that there is a big difference between the MZ and the DZ, right? And the next step, so this is the first part of the job. The next step will be to summarize these plots with just one number and to use that number to compute what is called heritability. All right? And this heritability will inform us on the part of this spatial organization in the brain that is due to the genetic part, so MZ versus DZ, and which is part from the environment. All right? So if we have a high value of heritability, then it means that this spatial organization is due to the genes. If not, it's due to some environmental factors. All right? And there is something more that we have not done yet, is to incorporate, I talked on the first slide of the white matter integrity, is to incorporate some information about the quality of the fiber. Right? So the quality of the fiber. On the image, we extracted the fibers, but using the same images, we also have some kind of information that informs us about the quality of the fibers. All right? And then when people get old and sick, like Alzheimer or that kind of problems, then the integrity of the fibers, so the white color comes from the fact that all these fibers, they have something around that helps the current to go through. And when this thing is deteriorating, basically the current cannot go through as fast as before, and then you end up with a lot of medical problems. All right? So we need to incorporate that information into the model. All right? But it's not difficult. It's just a way of adding some weight in that formula, basically. Right? All right. So this is what we have obtained, and I think, as a conclusion, we have developed a novel notion of data depth that can work on a space of unparameterized curves, and we call that the Tukey curve depth. It works by removing the parameterizations, and so it generalized in that sense the data depth that was defined for function, the functional data depth. It only exploits the geometry of the data. We have developed some properties of this new notion, as I explained, between 0 and 1, vanishing at infinity, similarity and variance. And we used that thing. So I presented the example of the brain imaging, but we have used this thing also to to do a pattern recognition or supervised learning. So yesterday we presented this MNIST data set where we had to find these numbers, these digits, right? So we applied that in that context, so it can work also. And we have this R package on the CRAN that you can use. We studied that until now for 68 brains. It took us days of computation on this HPC cluster of the Faculty of Science that I talked about yesterday. So, of course, we have now 20,000 brains, so this will be not enough, and we started to compute things on this big supercomputer in Australia, right? Using some parallelization of the code that is in the package. Right? So thank you for your attention. You have these references here if you're interested.